Okay, so the next speaker is Kamal Sharma, who is a researcher in the biophysics group at the School of Physics in the Georgia Institute of Technology in Atlanta, USA. And this talk will be about detailed sensitivity analysis of the OVVR human ventricular model in 1 and 2D tissues. Are you able to share your screen, Kamal?
I'm sorry, but there are two persons with the same name, so I don't know which one is to be enabled. I hope that is the same person. Okay, here we are.
Screen sharing has started, but we cannot hear you.
Kamal Sharma, please unmute your microphone.
The microphone is off.
Yeah, can you hear me now?
Yeah.
Can you hear me?
Yes, we can hear you.
Okay. Okay, so hello everybody. My name is Kamal Sharma and I'm a postdoc at Georgia Institute of Technology in Atlanta. First of all, before starting the presentation, I would like to thank Abuzar for making the WebGL library that I'm using for my research. And I'm very thankful to my professor Flavio Fenton as well for welcoming me to his family, to his academic family, and for being an excellent mentor to me. So for today's presentation, I'll talk about the sensitivity analysis of the OVVR human ventricle model in one-dimensional and two-dimensional tissue. So the OVVR model was developed in 2011, and it's based on experimental measurements taken from more than 100 undiseased human heart cells, and that's pretty impressive. It also shows the steady state measurements of action potential traces, which you can see here, which are taken at the room temperature. The model also reproduces the calcium ion rate dependence, which is here. They have used an AP clamp, where you can see the rate dependence of ICAL. And you can see that the experiment and simulation match really well. This model also reproduces the correct resting membrane potential, maximum upstroke potential, and maximum upstroke velocity within the error range, as you can see over here. And they have basically defined three kinds of cells: the epicardial cells, endocardial cells, and the M cells. Now, as seen before, the agreement of this model with the experiments is pretty good. You can see here that the APD rate dependence for every one of them, 30, 50, 70, and APD 90, matches really well with the experimental observations. And for the second one, this is the repolarization rate, where they have used the S1, S2 protocol, where S1 equals to 1000 milliseconds. Okay, so let's get to the mathematical part of it. The OVVR model basically contains 16 currents, as listed over here, and 41 state variables. So this is per cell. That means for every time step, you have to solve 41 ODEs. Now, this model is very complex. So what can we say if the model is very complex, and it also has some types of cells, like I mentioned before, epicardial, endocardial, and mid-myocardial. It also reproduces some of the basic experimental dynamics, as I've shown in the slide before. The question still stands: is one model with fixed parameters enough for predictive studies and fine dynamical mechanisms? Now, to answer that question, we have to understand the importance of uncertainty quantification, the sensitivity analysis, and the variability when evaluating computational methods. So I'll be just using the abbreviations. UQ involves empirical determining the uncertainty in model inputs, typically resulting from natural variability or measurement error, and then to calculate the resultant uncertainty in the model outputs. The sensitivity analysis involves calculating how uncertain is the model output and what are the sources of that uncertainty. The variability quantifies the difference within a species. So for example, for same kind of cells, you can have variability between different species or even in different individuals. So if a model has fixed parameters, then how does it take care of those variabilities? So this quantification is very important because it gives us a confidence to basically about the robustness of the model and the underlying uncertainties in case we want to use it for clinical purposes and for predicting arrhythmias. The variability explains differences in expressions, for example, race differences and how different individuals react to different drugs and different diseases. So basically the big problem here is the measuring and quantifying parameter sensitivity and variability in a highly nonlinear model can be computationally expensive. And especially when you're dealing with a very complex model such as the OVVR model, and that too, it's a single cell model. And when you are dealing with tissue, you have a lot of cells talking to each other. So the simulations become more computationally expensive. So let us do just a back of the envelope calculation of how much resources are required. First of all, the solution, as we have seen, the action potential is a very stiff solution. So it's slowly varying in certain areas and suddenly spikes. So you need to use a very small dt. We know that the number of ODs per cell is 41, and the number of cells simulated depends on the spatial resolutions, which is about 100 microns. So to simulate a strip of 5 centimeters, in 1 second, one requires to solve 2 to the power of 2 times 10 to the power of 10 ODEs. Now, if you want to go further and want to calculate the restitution plots, you need at least 20 beats to be close to the steady state, and then draw a curve, which will also require at least 20 different periods. So this comes out to be about 4 trillion ODEs. To test the sensitivity of the 40 conductances, the parameters in the OVVR model, we have to vary them to study the variability, and we can vary them from 0 to 2 in steps of 0.5. This will take this will come out to be 8.2 into 10 to the power of 14 ODEs. So basically, to perform a study for the effect of 2 conductances on a spiral wave dynamics, which is in 2 dimension or maybe in 3 dimension, for 2 dimensions, we have to solve 6.7 into 10 to the power of 15 ODEs. Now, luckily, our group has performed a study on the 100 model, which shows, if you look at this plot, you will see that the speed up direct scales with the number of processors used, which gives us an idea that if we use a lot of processors, then we can basically make the simulation faster. And GPUs are a perfect thing for that. So GPU is a cheap way now to support high performance computing with parallelization in a local machine. There are many options to program in GPU. We have NVIDIA CUDA, we have OpenCL, OpenACC, OpenGL, and the WebGL is a combination of JavaScript and GLSL talking to each other. Basically, CPU and GPU are talking to each other and giving instructions to each other. The advantages of WebGL are many, but some of them are, it allows a direct access to GPUs from a web browser, so you don't have to install any plugins or anything like that. It is now possible to run high performance parallel computing simulations over the web on a local PC or laptop independent of the operating system. So it doesn't matter if you have a PC, Mac, Linux, tablet, or a cell phone. You can use WebGL on that if it supports WebGL, that is. Now, what about supercomputers? So while using the supercomputers require creating and waiting for batches, and then you have to wait for the data transfer, then analyze the data and visualize the data. And if something has gone wrong, then all the effort has gone to waste. But in WebGL, by design, the calculations are performed on images, on the pixel values of the images, and you can basically just see what's happening in the simulation in real time, and you can interact with the parameters on the fly. So the library in our group was developed by Abuzar. This library is basically it handles all the complexity of the GPU simulation and all the plots in 2D and 3D, the beautiful plots I'll show you in a few minutes. And this code has been made free by him on his website. There's also a GitHub link. I haven't added here, but I will add it later. So the group has shown that the two articles have been published showing the use and capabilities of this library to handle complex interactive problems. So the examples that our group has shown that the GPU is very good in solving is, number one is the turbulent flow, second is the crystal growth, and then obviously the cardiac dynamics of a porcine ventricle. But let's get to the actually experiencing the stuff. So a WebGL code can calculate APD and CV restitutions if you're running it in one dimension. And basically, this is the interface you get when you click on this link. So this is an online link which you can click, and it will just open in a browser window. Can you guys see the browser window?
No,
okay.
We cannot see the browser. Maybe you only share the presentation and not on your desktop.
Okay. Just a second. Okay, can you see it now? Yeah. So this is basically how the go ahead. How the interface looks like. And in this interface, you can basically do a lot of things. You can choose a one-dimensional tissue length. You can change the spatial and temporal resolution. You can change the simulation site, measure pixels for voltage, APD, CV, and other restitution plots. You can also modify all the 64 parameters in real time on the fly without even resetting the system. And you can also change the cell types. So let's just quickly go through this. This is the system, and there's a canvas where you can see the cells. So first of all, we can define the parameters, which is the resolution. Because this is a one-dimensional simulation, we will set the number of pixels in the y direction to be 1. And we can set delta t to be 0.8. The next thing is we can play with the pacemaker. We can make it active, and we can apply a pacing period of 500 milliseconds. And because we want to pace it from the left end, we will choose 0x coordinate for that. All right, so let's play this. I can also make the probe visible. So simulation is basically measuring everything at this location, at the center of the cell, and you can also change this. Now you can see that there are APDs going on, action potentials, and then the total current you can see over here. But let me show you another functionality. We have APD measurement. So if you check this box, you can basically see, let me make it bigger. So here you can basically see what is the BCL, the DI, the APD, and the conduction velocity. So let me quickly show you how it can generate. Let's go to mid myocardium. So this is how we generate the restitution plots. You make it a little bit faster. So this can actually go really fast. And then you can start reducing the pacing period. Let's go to 400. And now let's go to 350. So as you've seen, there's a dramatic change that happened here. And if I slow it down, you'll be able to see the presence of alternance here. You have alternating action potentials. The alternance can also be seen in the current over here. And you can change this current, you can change this plot to show any kinds of current that you want. Okay, so back to the presentation. Okay, so this was one-dimensional APD and CV restitution calculation. And we have done this for all the current parameters. And we have basically found 4 classes of this. So when you change the conductance of sodium from 0.5 to 2, there is no change in either conduction velocity or the APD. For Ks, there is a lot of change in APD, but no change in the conduction velocity. Then there are cases where you have effects on both APD and the CV. And finally, this case where you have an effect on the CV, but not on the APD. So now all this can be basically written in a script, and it can run very fast, and this analysis can finish in a couple of minutes. Okay, so we have also worked on two-dimensional dynamics. And instead of showing you two-dimensional dynamics, let me show you a three-dimensional model of the heart that we recently developed. So let's reduce the frame rate because it's so fast that I cannot respond to create. I can't respond as fast. Okay, so we excited.
Sorry for interrupt. I'm asking you to get to the conclusions as fast as you can, so we can have room for at least one question.
Can you give me 2 minutes?
Okay, thank you.
Yeah. So as you can see, we can increase the speed here, and basically you can generate all kinds of waves. Okay. So you can change everything that we have discussed for the one dimension here as well. And if you made a code out of it, you can basically automate the production of these phase portraits where you can see what kind of spiral waves are there in different parameter ranges. So here you can see the spirals are breaking up, whereas in other parts, the spirals are not breaking up and they're pretty stable. And sometimes they meander off the domain. Again, here we have plotted the same thing, but this time one is for current and the other is for time coefficients. And you can see how as C tau d increases, you get wave breakup and finally a lot of chaos. You can also see EADs develop in this, but let me move ahead. Don't have that much time. Okay, so this is an important result. If we block the IKR current, we observe EADs. And in this case, for the first figure, you can see that we have, if you have 80% IKR blockage, you have long-lasting EADs. Now, we can also talk about dimensionality for single cell, one dimension, and two-dimensional tissues. So for one dimension and single cell, there are EADs, but they die out pretty soon. But for 2 dimensions, we have early after depolarizations, and basically they last for a long time. And finally, I wanted to show you, this is the last video, how you can basically create arrhythmias and reentrant EADs in a 2-dimensional tissue by just all produced by one single stimulus. Okay, so this movie shows just one stimulus from the corner, which is the lower left corner, and the rest of the dynamics originate from that single stimulus, which goes on to spiral breakup and then generation of arrhythmias. For conclusion, we have demonstrated a new spiral wave behavior with EAD activations on the back of the wave that can actually propagate and destabilize the wave, and we have used WebGL for these techniques, which is very fast, and as you can see, highly interactive, and you can change parameters in real time. And to the best of our knowledge, we are the first one to demonstrate the example of re-entry initiation by a single stimulus in homogeneous media. I would like to thank NIH and NSF for their support, and also thanks to my research group. Thank you.
Okay, thank you, Kamal, for your great presentation. We have time for one quick question. As usual, please raise your hand or write a message. If nobody has, actually, I have one. And my question is, how do you think that what do you think there are the limitations of using GPU platforms versus CPU one? I mean, usually the differences between the two are finding a trade-off between memory.
You're right. Right.
Sorry.
So that's
Okay,
a good question. I can say that the plot that I've shown you, which says that the speed up is basically scales with the number of processors. In a typical graphical, in a graphic card, you have thousands of GPUs, which can basically parallelly calculate everything, while a CPU can only handle as many cores as it has. So a CPU basically redistributes its resources. Whereas in GPU, you have a lot of resources to do simple calculations. It's very good for systems like this, where you have a high complexity, but you still have a linear dependence of speed on the number of processors required.
okay.
A challenge would be because it's not computationally, it's very tough to program WebGL. So that's a very steep learning curve. It takes a lot of time to learn it, and sometimes you might even have to hire or some person from computational background.